Today we will discuss about how the glycans plays the role in the immune response in the normal state, that is in the homeostasis, and how this alteration of this glycans on the immune cells will cause immunosuppression in the cancerous state. So let us start with the homeostasis. So this is the T cell development. It is in the DN3 stage, and when it comes into DN4 stage, it can interact with this. It can regulate this TCR selection with the help of this expression of glycan when it converted from DN4 stage to DP stage, and this expression of glycans will then helps them to interact with the will help them to interact with the specific any foreign antigens that are present within the body against which the antigen response has been triggered, or the other function of these glycans that are present on this stage, this DP stage of the T cells, is to help in the regulation of this C cell traffic. So this C cell traffic will there are two factors on which this T cell traffic will depend. First, it will depend on the signals that it has received regarding the foreign antigens present within the body, that it can receive with the help of this T cell activation, either via mature dendritic cells, or by directly detecting the antigens that are present within the body. So this way it will regulate the T cell traffic, and during this process the glycan also plays an important role. And when these glycans come in close with the other neighboring immune cells such as CD25 and CTL4A, it will start producing less complex and glycan structures that can be seen here in this TCR. The TCR is the form of this T cells, and this CD25 is another immune cells, and this CTL4A is the cells. We can say the CTL4A is also another variant of the T cells. And so this is how the functioning in the normal case occurs. And when it in the case of cancerous cells, what happens is that these T cells will not be able to directly interact with the tumor cells because of the aberrant glycosylation takes place within the tumor cells. One of those glycosylation is that there are excess sialic acid that are present within these cancerous cells, and this excess sialic acid will form a shield and prevent these T cells to directly interact with the tumor cells. And at the same time, the tumor microenvironment got changes, and because of the metabolic changes that are occurring within the tumor cells that we have discussed in the previous videos, such as decrease in the pH in the tumor microenvironment, and also we can see that there is a change in the glycosylation profile compared to the normal homeostasis profile, and that will cause various changes such as this CD8 T cells apoptosis will not be able to function properly, and this increase in the immune checkpoint inhibitor. We know that in order to activate T cells, there are two kinds of signals are required. First one is that the foreign antigen signal, and second one is also along with the foreign antigen signal there is a confirmation signal, we can say that, and that is usually been inhibited by the cancerous tumor cells. And so this is another way by which the immune suppression occurs within the cancer cells. And other way is that there is an increase threshold of this TCR activation. In this case, we can see that this TCR will activate when it sees the foreign antigen and at a certain level, and that certain level will be increased to a very high extent when in the case of tumor cells. So when the tumor cells, in the case of cancerous cells, even when it sees the foreign antigens, it will not be able to trigger immune response to a very high level compared to the normal homeostasis case. And another thing that is mentioned here is about this drag expansion. This drag expansion is, you can say, is related to the galectin inhibition. So galectin is the one of the signaling pathway that will trigger apoptosis. So that galectin activation will get disrupted or inhibited because of this drag expansion, so that this galectin related apoptosis will not be able to trigger. So these and the final panel in the bottom shows the various immunotherapy targeting agent that can be used. So one of the targeting agent that is shown here is about this activation, is about, we can say, activation of this immune checkpoint through this enhanced Glycan structure that can be shown here, that is shown here. So, and other way is about this CAR T cell Siglec 7/9 using Siglec 7/9 antigen. So this is usually more aberrantly found in the tumor cells. This can be used as an antigen in the CAR T cells that can use to target those tumors that have this Siglec 7/9 antigen. And this about this PDL1 is the one of the immune checkpoint inhibitor. So this thing can be prevented from inhibition, or it can be activated through the glycosylation that are expressed on this PDL1 in the case of cancerous patient. So this glycans can be used as a signature, and that can be used to activate the T cells. Other thing is about this resistance to apoptosis. So this resistance to apoptosis can be prevented using this increase in the amount of alpha-2,6-sialic acid. So it has been found that increase in the amount of alpha-2,6-sialic acids will inhibit this galectin-1 related galectin-1 that usually prevent this galectin-1 related antigen that usually prevents the activation of apoptosis within the tumor cells. So this thing can be inhibited by the alpha-2,6-sialic acid, increasing the alpha-2,6-sialic acid. So these are some of the approaches. And another approach that is mentioned here is the increased IFN secretion. So this is one kind of cytokines that are released whenever there is a harmful things detected by the body. So this thing can be increased. So it will also leads to decrease in the decrease in the tumors within the patients. And this is again it is mentioned here is about the checkpoint inhibitor blockage. So the glycans can be used, or glycans can be engineered in a way that it will block those substance, or it will block those substance that is used to inhibit the checkpoints within the tumors, in the tumor cells, or in the tumor microenvironment. And this PDL1 is not about checkpoint inhibitor that I have mentioned wrongly previously. This is about the activation of apoptosis. So this thing can be activated through with the help of this glycans that can trigger this PDL1 activation. So these are some of the immunotherapies possibilities that can be used, or these immunotherapies are related to the glycoengineering, that is to engineer the glycans that are expressed on the T cells to target the tumor cells. So if you need to understand more about this T cell glycoengineering, that can be used as another alternative immunotherapeutic strategy to target against cancer. You can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.